Assalamu alaikum everyone. In this video lecture, we will be discussing some tricky MCQs from the new syllabus of O-Level Biology 5090. In a length of DNA, 20% of the bases were T. What is the percentage of the base G in this length of DNA? So guys, they have given the information that in a length of DNA or in a piece of DNA, T bases are 20%. We need to figure out the percentage of base G. So how do we do that? You need to know that adenine or A pairs with T or thymine in DNA, right? So because A pairs with T, the number of bases, the number of T should be equal to number of A, which means that A should also be 20%. If T is 20%, A should also be 20%. And because C pairs with G, so percentage of C should also be equal to the percentage of G. Because the total percentage of bases in the DNA is 100, what we can do is that we can minus 40% from 100, right? To get the answer that is 60%, which is C plus G, right? That's because if thymine is 20%, adenine is also 20%. This makes up the 40% of the DNA. So the remaining 60% of the DNA will include C and G. So C plus G is equals to 30%. Because C is equal to G, this means that C is 30% and G is also 30%. So the answer is C, 30%. Let's move on to MCQ number 2. The graph shows the growth curve for a population of bacteria which identifies each phase shown in the graph. The table identifies each phase shown in the graph, which row is correct. So guys, this is the population curve of bacteria, and there are some important phases in the population curve of the bacteria. So initially, when the bacteria start to grow, their growth is slow, and this is known as the lag phase. Whenever bacterial growth is slow, initially it's called the lag phase. Then bacterial growth is very rapid because the bacteria, they adapt to the new environment. Initially, bacteria are added to the nutrient medium, they're added to the new environment, so it takes time for them to adapt. So initially they grow slowly, and that is the lag phase. When they become adapted to the environment, they grow very rapidly, and this phase is known as the log phase or exponential phase. Then there's a phase in which the number of the bacteria become constant. This phase is known as the stationary phase. And then when the nutrients are depleting, what happens is that the number of the bacteria decrease because bacteria start dying, and this is known as the death phase or decline phase. So you need to know that the first phase, the phase P, is the lag phase. Then R is the stationary phase. Exponential or log phase is phase Q, and death phase or decline phase is phase S. The answer is B. Let's move on to MCQ number 3. Two animals have an identical sequence of amino acids in one of the proteins found in their cells. What does this indicate about these animals? So guys, if there are two different animals and if they have identical sequence of amino acids in one of the proteins found in their cells, this means that those two animals are closely related. Now, close relation does not mean that they must have the same genus. They could have the same family, they could have the same phylum maybe, or they could have the same order maybe, right? So it's not necessary that they must have the same genus. They simply are closely related. They can have the same genus, they can have the same family. So the answer should be that they share an ancestor, because if they're closely related, they have a common ancestor from which they have evolved. The answer is D. Let's discuss MCQ number 4. Which diagram shows a flower from a monocotyledon? So guys, always remember this point, that whenever you want to recognize a flower from a monocot, just look at the floral leaves. If the leaves are thin and have parallel veins, then that is the flower from the monocot. The answer is D. Let's move on to MCQ number 5. What is the function of the lymphatic system? Guys, lymphatic system is a system which consists of tubes which carry a fluid called lymph. Just like we have blood vessels which carry blood, we have lymph vessels which carry lymph, or we have lymphatic system containing lymph vessels or lymphatic vessels which carry lymph. So one of the example of lymphatic vessel or lymph vessel is lacteal, that is found in the villi of the small intestine. So lacteal is a lymph vessel which contains a fluid called lymph, and as you all know that the function of the lacteal is to transport fats, right, and the fluid, right? So circulation of body fluids is the general function of the lymphatic system. When the blood flows into the tissue, the fluid leaks out of the capillary, and this fluid is called the tissue fluid. Majority of this fluid returns back to the blood capillary, but some of the fluid stays in the tissue, and that fluid is evacuated by the lymph vessels. So lymph vessels are also a system of vessels in the body which are involved in the circulation of body fluids. The answer is C. MCQ number 6. What holds the water molecules together during the transpirational pull in the xylem? So guys, when the water is traveling up the xylem vessel, the water is pulled by the transpirational pull. That is the force generated by transpiration. But what holds water molecules together are the forces between the water molecules. So for example, if this water molecule is pulled by the transpirational pull, that is the force pulling the water up the xylem, the remaining water will also be pulled. And how is that so? This water molecule will pull the water molecule beneath it. And this is because of the forces between the water molecules. These are basically the intermolecular forces. And the forces between similar molecules are known as cohesive forces or cohesion. So if we say that there are forces between the water molecules, we can say that this is cohesion. Why? Because cohesion are the forces between similar molecules like the water molecules. So the answer is B. Cohesion is a force which holds water molecules when the water is traveling up the xylem vessel by transpirational pull. Let's move on to MCQ number 7. Human insulin is a protein that can be made by genetically engineered bacteria. This involves the transfer of genetic information to bacteria. The diagrams show cells and parts of the cells involved in this process. What is the correct order of transfer of genetic information? So guys, what we do when we synthesize human insulin using genetic engineering or genetically engineered bacteria, what we simply do, we take the human cell, we remove the insulin gene from the human cell, or we can simply say that we take out the DNA from the human cell and we cut out the insulin gene from the human DNA, and then we cut the plasmid, right? And then we insert the insulin gene into the plasmid, then we reintroduce the plasmid into the bacterial cell, and what happens is that the gene on the plasmid, the insulin gene on the plasmid, is then expressed, right, and the protein is synthesized on the ribosome. So what is the sequence? The sequence is from the human cell to the plasmid, from the plasmid to the bacterial cell, and from the bacterial cell to the ribosome. Ribosome is the last one because ribosome is the site of protein synthesis, right? And insulin is a protein which is synthesized at the ribosome. So the answer is A. Thank you so much for watching.